Welcome to the Innovative Science Accelerator program funded by the NIDDK. My name is Dr. Richard McIndoe, and I'm the director of the ISAC Coordinating Unit at Augusta University. Today, we have the pleasure of welcoming Dr. Oleg Denisenko, an ISAC Award recipient. He'll present his work developing a new assay called the Epigenetic Visualization Assay, or EVA. This new assay will allow investigators to interrogate epigenetic state of a specific gene of interest in a single cell.
My name is Oleg Denisenko. I'm at the University of Washington in Seattle. I'll talk about epigenetic visualization assay that we developed to study epigenetic of individual genes in single cells. This method is based on proximity reaction between a phosphate group attached to an oligo and the enzyme alkaline phosphatase. Gene is recognized by a series of oligos that are hybridized to the locus, and each oligo has an extension. This extension binds to a red oligo with the phosphate group at 5' end and the green oligo. If there is an epigenetic mark at the gene of interest, shown here in purple, this mark is recognized by a specific antibody, and secondary antibody is conjugated to alkaline phosphatase. And if there is a proximity, phosphate group is removed, and this oligo is resistant to lambda hexanuclease, which is dependent on the 5' phosphate group. If there was no epigenetic mark at the gene, the red oligo is degraded up to the gap, and the green color is gone. So the intensity of or density of epigenetic mark at the locus is measured as a ratio of green to red signals. So we tested this design in the model system in human cells that contain one copy of HIV provirus, and the probe contains, in this case, 20 oligos to the 5' end of the viral genome. So in this figure, you can see example of representative cells where you can see red signal, one per nucleus, and the green signal, which is the measurement for the 5-MC DNA methylation intensity at the locus. And in the control experiment, no antibodies were added. And you can see that there is still red dot, one per cell, and there is no green signal. So the intensity of green and red signals were measured, and the ratios are shown in the graph. So there is wide distribution in intensity of DNA methylation in the locus. In the control, the ratio is pretty low. So when we measured multiple cells, we can see the efficiency of this method. So in example shown in the image top right, you can see that all cells in this area reveal HIV EVA signal. So all cells contain either 2 dots per nucleus or 1 dot per nucleus. Analysis of hundreds of cells show that at least 90% of cells reveal HIV EVA signal. And the majority of cells had 1 dot per nucleus, and about half of that number had 2 dots per nucleus. And the 2 dots correspond to cells in the process of mitosis. And the intensity of HIV signal was pretty high compared to background. So the question is, can we use this method to measure changes in DNA methylation at the locus of interest? So in this experiment, we used an inhibitor of DNA methylation, decitabine, and we treated cells with this inhibitor for 48 hours and compared 5-MC signal at HIV in treated cells versus untreated cells. And you can see that in treated cells, the signal is substantially lower, as expected. In the control experiment, we used another method, alternative method, to measure DNA methylation in bulk DNA that was purified from cells, same cells treated with DAC or not treated. And the method was MED-IP, methylated DNA immunoprecipitation. And these results confirmed findings that were visualized by EVA method. EVA method can be also used to examine histone modifications at loci of interest. So in this experiment, we used a probe shown here in green to ribosomal DNA locus. So remind you, ribosomal DNA encodes ribosomal RNA. So the primary transcript is processed to 18S, 5.8S, and 28S transcripts that are used to build ribosomes. And there are 300 on average copies of this gene per human genome. So when we use EVA probe to this locus, we can visualize multiple dots as expected. And these genes are clustered. That's why we see dots of irregular size. So the antibodies in EVA were to acetylated lysine 9 on histone H3. So you can see that green color corresponds to some of the ribosomal DNA clusters, not all of them, which indicates that not all clusters are transcribed in this particular cell. In the control experiment, where no antibodies were used, we can still see red dots, but no green signals. So next, we combined EVA with RNA FISH for simultaneous analysis of epigenetic marks at the locus and the transcriptional status of the gene. So the EVA probe was again to the same HIV locus, and RNA FISH probe was to the GAG pole region of the virus. So the cells that we use contain this HIV virus silenced. So in order to induce transcription, cells need to be treated with a mitogen when we use the TPA in this case. And after 8 hours of treatment, 10% of cells show RNA signal of HIV virus. So in this example of positive cell, RNA positive cells, you can see that we can measure epigenetic mark, in this case 5-MC signal at HIV locus, and RNA signals, which corresponds to nascent transcripts. So EVA can be used to measure both transcription and epigenetic status of the gene. Here is another example of combined FISH, RNA FISH and EVA. So we used probe to imprinted gene exist. So EVA probe was to the beginning of the gene, and RNA probe was to the end of the exon 1. So to remind you, this gene is expressed from one allele only in female cell lines. And the allele that is expressed is hypomethylated, and the second allele, which is not expressed, is hypermethylated. methylated. So we use this probe with these two probes to detect RNA and 5-MC signals at the exist locus in female cell line. So the probe can detect two loci in each nucleus, and only one of those loci contains intense 5-MC signal, and the signal of 5-MC at the second locus is second allele is lower, and the hypermethylated allele overlaps with the RNA fish signal, which is exactly what we would expect. In the control experiment, sorry for that, we used no lambda exonuclease treatment, and both alleles have intense green signal. And when we quantitated green and red signals in these cells, and the results are shown in the graph. So you can see that the 5-MC signal at the RNA negative alleles are always higher than at the RNA positive alleles. So this result shows that EVA can be used for RNA transcription analysis and analysis of epigenetic status of the gene, and it allows to do selective analysis of individual alleles. So as a summary, we developed a method for epigenetic analysis of individual gene alleles in single cells. Epigenetic analysis can be combined with RNA transcription analysis, and EVA can be multiplexed with for analysis of multiple genes per cell. So currently, in the current EVA protocol, it's just only one gene at a time that can be examined, but it can be multiplexed for study of multiple genes. So this method development is a result of team effort. I would like to thank these people that helped to accomplish this study, and thank you very much.
I'd like to thank Dr. Denisenko for taking the time to share this new technique with us. If you have an exceptionally innovative, high-risk, high-reward idea that has the potential to lead to groundbreaking or paradigm-shifting results, ISAC may be the place for you. Applicants can request up to $100,000 for 1 year, and the projects must be relevant to the kidney, urology, and hematology research communities. If this program is of interest to you, please visit us at www.isac-kuh.org. Thank you.